Hey guys, and welcome to the first of many that we'll be going over remotely learning about some of the content in this unit. So in terms of what you're going to be learning about today, there's 5 big ideas. One is going to be that when we do chemical reactions, we don't lose any atoms. Matter is not lost, but the energy within those chemical molecules is going to be transformed and changed. The second is that the energy stored within chemical bonds is dependent on 3 key ideas: electronegativity, bond type, and molecular size. When we look at types of chemical reactions, exergonic reactions, which we can see right here, are reactions in which we're going to reduce the amount of stored energy within our bonds. And the last 2 ideas have to do with the role of enzymes. Many living things use enzymes, which are specialized proteins that can help reduce the amount of activation energy within a chemical reaction, making our efficiency increase. We get more money out of the reaction, so to speak, and that reaction rates are really determined by enzyme substrate concentrations, the relative amount of substrates and enzymes. So when we look at a chemical reaction, what we're going to be looking at this unit is specifically heterotrophic metabolic rates. Heterotrophs are organisms that use other energy to provide fuel for them to survive. Plants, on the other hand, are autotrophs. There's 3 key reasons as to why these processes are really important for living organisms. One is going to be releasing heat for body warmth. This is especially important for, say, warm-blooded organisms. It provides immune support, and it allows chemical reactions to occur. Often an increased temperature in the body is a way for us to kill off bacteria if we're sick. The second is that a lot of this energy that we release is going to allow our cells to make ATP. And as we talked about last unit, phosphates are incredibly important for cells maintaining homeostasis. And the last aspect is that chemical reactions allow us to recycle matter back into the environment. A lot of waste that we generate from metabolic processes go into the soil, water, and air, allowing other organisms to survive. If an organism runs out of energy, it will die. So it's constantly occurring within our bodies every single second. There's 2 types of chemicals within a reaction. One are reactants and one are products. In our chemical reaction of cellular respiration, we're going to be reacting 1 molecule of glucose with 6 molecules of oxygen. Because oxygen is super electronegative, it's going to help cleave apart that glucose with the help of enzymes, and that's going to release a lot of energy as a result. We're going to make a lot of ATP. The products, which are molecules that we make from our chemical reaction, are going to be 6 molecules of CO2 and 6 molecules of H2O. They are considered waste products, and we'll get into that in a little bit. Now, in terms of chemical reactions, there is a very important law that we need to first establish and understand within why these processes are working, and that is the law of conservation of matter. And what that means is when we look at our products and our reactants, the amount of carbon and oxygen and hydrogen in terms of the number of atoms or the mass will always be the same before and after. We can't destroy matter, we just rearrange it in terms of the types of bonds and strength of those chemical bonds. When we look at this chemical reaction, we have 6 carbons and 6 carbons in our products and reactants. We have 18 in our products and our reactants. I'm realizing these should be flipped. We have 12 atoms of hydrogen and 12 atoms of hydrogen. So we're not really losing or gaining anything in the process. When we apply this to mass and we think about chemically balanced reactions, which you'll learn about in chemistry next year, if I have 180 grams of glucose, I'm going to need 192 grams of oxygen to react that. Again, we'll learn about this in chemistry next year, giving me a total mass of 372 before. And when I look at the mass of carbon dioxide and water produced as a result of that reaction, it also will be 372. So whatever reaction we are going to do in all of our labs, there should be no products that are unexplained or are surprising. What we put into the reaction is what we're going to get out of the reaction in terms of mass and atoms. Now, this is not going to be true for the energy. And what we need to know about energy within chemical molecules is the amount of energy and movement that electrons have within a particular atom and within a particular molecule is going to determine what that reaction does in terms of releasing or absorbing energy. And the movement of electrons is going to be dependent upon how far away they are from one or more nucleuses and how many electrons are in that outer valence shell. The further away electrons are from a nucleus, the more energy they will have, which means they can do more work when we either break the bonds or set them free. So rules of energy. When we talk about these reactions, we're going to be talking about whether the products or the reactants store more energy. And energy, as I just explained, is going to be dependent upon the movement of electrons. But there's a couple of rules that we need to establish as to whether or not chemical reactions are going to be releasing energy or absorbing it. The first is this: bonds between atoms with smaller electronegativity differences store more energy. When I look at oxygen versus carbon, both bonded to hydrogens, these electrons are going to be further away from the nucleuses of carbon and hydrogen because the difference in electronegativity scores are smaller. This means neither carbon nor hydrogen is stealing or grabbing onto those electrons. They should have more movement as a result. Alternatively, oxygen, it's going to be pretty grabby with those electrons, so it's not going to have these electrons storing a lot of energy. The movement of electron, again, determines their energy, so polar bonds have less movement and have less energy. The second rule is double bonds tend to have less energy than single bonds. The double bond means that there's twice as many electrons in the same space as a single bond. So notice I have 1, 2, 3, 4 electrons. All of these negatively charged electrons are stored in this area, but they all repel each other because of that negative charge. That means they're constantly fighting for a smaller amount of space. They're going to have less movement, less energy, and hence this bond will store less energy. And the last idea is bigger molecules have more energy. Bigger molecules have more opportunities to have either of these types of bonds, polar or nonpolar, single or double. And this is why we often see fat having more calories than sugar or proteins. It's because there's lots of single bonds that are all carbon and hydrogens, while glucose, for example, is a lot of oxygen to hydrogen or double bonds. So what does this mean? When we look at cellular respiration in yeast or our cells, we undergo a reaction that's known as an exergonic reaction. What this means is that the energy stored between bonds is decreasing in energy during the reaction, meaning we're going to release light, heat, or sound as a result. The energy that's being stored in our reactants is going to be released in some form of energy, and the products as a result will have less energy. Now, when we look at this graph, which is a really important graph for you to copy in your notes, we're talking about free energy stored within reactants and products. When an environment is filled with molecules that are low in energy, that means they often have double or polar bonds. And this means that the environment is pretty entropic or disorderly. Our ability to do work is pretty limited because all of the energy has been lost as heat or sound or some other product. And the products themselves don't have a lot of energy stored in their electrons. This is going to demonstrate a law that you learn next year in chemistry, which is the second law of thermodynamics. As reactions go from orderly to disorderly, high energy to low energy, it tends to be a system that is more chaotic, with less available energy, and it can do less work as a result. And this is because all of the things that accumulate in this product or in this environment are not really able to do work, right? You can burn a fire with a lot of wood, but at the end of the wood or the fire, there's just ashes, and those ashes have less stored energy in their molecules because it's been given off as heat and light and sound. You can't start a fire with the ashes. You fill your car with gasoline, the gasoline is reacted in your engine, the car exhaust can't be recollected to make your car run further. You eat a chicken McNugget dinner, you digest all of that food, the waste products can't be reabsorbed to make your body do more work. So we have to talk about how we can make these reactions occur. And the first idea is going to be related to breaking the bonds, and this is called activation energy. When chemical reactants or inputs are high energy, they tend to be less stable. They tend to have a lot of stored energy, but they still need those initial chemical bonds broken. Note in this reaction how the CH group goes from a state of high to low electron energy because we're adding this very electronegative molecule. So the energy that we need to break up the bonds between molecules in a reactant is called the activation energy. This energy is lost in terms of the amount of energy we gain from breaking bonds, which makes our reaction efficiency less. Another way of thinking about it is, how much do I have to spend on lemons to make lemonade? If I spend $5 to buy some lemons, but then I sell my lemonade for $10, my net profits are going to only be $5. So if I think about the change in reaction, this blue line is going to be my activation energy. This is the energy I have to invest to break bonds between molecules. This is like lighting a match to start a fire, or buying some lemons to make lemonade. You're making an investment. This is increasing the instability of the molecules that are in our reactants, which tend to make them a little bit more reactive. When I look at this red line, this is going to be looking at the difference between my reactants and products. This is the amount of gross energy I would be releasing. However, when I then subtract this blue line, the purple line is actually what I gain. Because I had to invest all of this energy, my net profits are only going to be the purple line in terms of energy that I could release to the environment or use to do work. This means that my reaction isn't that efficient. So again, being able to break bonds is a really important key to making our exergonic reactions occur, but it does take away from the efficiency of our reactions. So this is why we use enzymes. Enzymes are a tertiary structure protein with specific shapes that enable them to interact with substrates. So here's an enzyme that's reacting with a substrate. It's going to zoom in over here in a second. Substrate is going in, and this enzyme is reacting with it. This is almost always due to a positive-negative interaction between amino acids. This is an enzyme that's making DNA. This is an enzyme that's putting two molecules together. So all of these are due to amino acids being positive and negatively charged that can interact with some other molecule. Now, when we look at a substrate binding to the active site within an enzyme, there's two interactions that occur between the substrate and the enzyme. The first is the catalytic site. The catalytic site is going to be embedded with highly polar amino acids that react with charged regions of the substrate, sort of chemically helping pull them apart. It's almost like melting butter in a pan. It's reacting with the substrate in a way that it's going to physically and chemically make them not be able to retain their initial state. Think of it as dissolving something in a lot of acid. The second part of the active site is going to be the binding site. This is embedded with, again, positive and negative amino acids, but it's going to be physically moving the substrate to assist in the process. So it's almost physically splitting it up, like cutting a grilled cheese in half. And what we see is this enzyme is going to move once the substrate fits in the active site. And this physical movement alongside the binding site and the catalytic site are going to help make this molecule split apart. And the end result is this. When we look at the amount of activation energy needed to split up our molecules, enzymes, because of that physical and chemical structure, are going to make our net energy products way more efficient. Note that the red lines are the same between my reactants and products, but the purple line is increasingly much higher, meaning my efficiency rate, my losses are a lot smaller. And this is due to the physical and chemical properties of the protein. So I think that's going to be enough for today. If I go back and look at the initial objectives, I think things that we talked about today are the following. We talked about the fact that matter is not lost nor gained in a reaction, but energy can be. The energy stored in chemical bonds is dependent on three key ideas: electronegativity, bond type, and molecular size. Exergonic reactions are going to be reactions in where the products have less stored energy than the reactants. And we also learned that enzymes are specialized proteins that can help reduce the activation energy of reactions, making them more efficient. We'll talk about this tomorrow in class. Come in with questions, and I hope you have a good rest of your